Good morning, everybody. Welcome to a beautiful Chautauqua morning. And well, it'll be beautiful. Give it some time. I'm Bob Spiritus. I coordinate these talks. Our president, Bob Hopper's in the back. We welcome you to the science group presentation on Wednesday morning. We're here every Wednesday morning, same time. You can come in person, you can Zoom in, somehow join us if you can, whenever you can. Today, we're happy to have Dr. Wigdahl-Perry, who will be Dr. Courtney Wigdahl-Perry, who's an associate professor in the biology department at SUNY Fredonia and the program coordinator of the environmental science major program. And I guess I've got some competition out there from a truck, but can you all hear me all right? Yeah, okay, well, we'll have to talk over the truck. She joined the faculty in Fredonia in 2014 after getting her PhD from the University of Maine in ecology and environmental sciences. She also holds a BS in biology with an environmental science concentration and an MS in biology aquatic science. She focuses on the understanding of algae and water quality. Her research program studies how these valuable systems respond to different kinds of environmental change in the past and present. She's here with us today, courtesy of Mark Wenzler and who helps us with and he's the head, as you know, of the Chautauqua Climate Initiative. So we thank Mark and we thank you for coming. And I think without further ado, we're going to ask Dr. Courtney Wigdahl-Perry to come and give her presentation.
Thank you very much. Good morning. My time going. Excellent. Well, it is wonderful to be here as always. I am a scientist at heart, but I really enjoy how the research I perform connects so much to the community. So it's great to see so many faces here that are interested in learning more about what's happening right in your own backyard. So what we're going to do today is talk about bloom basics and provide you some information on what controls algae growth. And I want to make sure we spend time today talking about how scientists study this problem, right? And dig in a little bit more to the methods. And one of the reasons why I wanted to make that a central theme of my talk today is that very often you hear a lot of these terms thrown about when we're talking about the very nitty-gritty research data, and we don't always have the time to sort of walk through why scientists make the choices they do, why having these different tools in place is really helpful, and why there's not one answer to the question, right? So I want to make sure we spend time on that today. And then for the last portion of the talk, we'll focus on Chautauqua Lake specifically, what sort of the state of bloom affairs is, and what my team is doing. And we'll have more time to talk about those research activities in the Q&A as well. So a lot of this may be review or background information for some of you, but it's always good to have everyone on the same page. So to start off with, harmful algal blooms are considered an excessive growth of algae or cyanobacteria, and to the point where this excessive growth causes nuisance problems or negative effects on the lake ecosystem or marine ecosystem, and or how people interface with that system, right? The human uses. I've noted here that I'm separating algae from cyanobacteria. Often these groups, these organisms are lumped together because functionally they perform a very similar role. But in reality, they're two very distinct biological groups. Cyanobacteria, or the blue-green algae, have a much longer evolutionary story. They've been around for much, much longer than other types of algae. And so they have some very interesting, cool traits. And they operate a little bit differently. Okay, so sometimes you'll hear those terms used interchangeably, but just so you know, there are some differences there in terms of what causes them to grow and some of the features that they do have. Additionally, I've noted that there are algae that cause harmful blooms in the ocean and cyanobacteria that are responsible for the freshwater blooms. There's actually the red tides are a whole different group of organisms that occur in the ocean called dinoflagellates, and they're algae, they're true algae. And cyanobacteria are the primary culprit in our freshwater systems. You may even hear them called cyanohabs or CHABs to differentiate them for the freshwater lakes. So we'll use these terms interchangeably, but just so you know, there's a difference there as well. These bloom events, when they happen, have a number of different negative implications, right? I tend to group them into two categories where we think about how the lake status affects people and then how it affects the blooms affect the lake's health itself. And so in terms of humans, one of the biggest things that we're worried about is the human health implications, because cyanobacteria and the marine counterparts as well can produce toxins, neurotoxins, liver toxins, a whole suite of compounds that can be damaging to humans, pets, livestock, right, and other organisms that come in contact with them. And so the public health component is often what we're concerned about for good reason. Bouncing off of that, the recreational access can be limited if there's a bloom event because of that public health concern. And also for drinking water sources, if there are cyanobacteria toxins in those sources, they are not easy to remove from the water, right? You can't boil it. You just have to wait it out most of the time, okay? So we have some real public health concerns from those standpoints, and the aesthetic value and the recreational limitations can also affect our economic impact, right? We have many, many businesses around the area, lots of tourism, boating, recreational fishing, major industry is happening around Chautauqua Lake. And the lake water quality affects how many dollars come into our communities. So there's a number of layers there in terms of why we care about HABs. In terms of the lake itself and the ecosystem health, after a bloom event occurs, that biomass, all that stuff, stays in the lake and sinks down to the bottom and is decomposed by other bacteria. That decay process uses up oxygen. And so we can have really, really low oxygen values after a bloom event, and that can lead to potentially massive fish kills, right? Which is, as we know from the world-class muskie and bass fisheries that we have here at Chautauqua Lake, is a very real concern from the lake health standpoint, but also in terms of our human uses. This sort of stress, these sorts of changes in the food web structure mean that at the base of the food web, where energy comes in and is changed into sugars by these green little producers, the nutritional quality of that stage of the food web is very different between cyanobacteria in bloom state and other kinds of algae, right? It's sort of the difference between having like a nice healthy salad over here with this variety of algae and like a Big Mac. It's not easy to eat. There's not a lot of nutritional value. You can do it, but you have to work pretty hard, right? So there are some different nutritional values. And so these pressures at the base of the food web can ultimately lead to shifts in the food web structure overall, okay? So again, sort of two categories in terms of things that we're interested in for bloom issues and why people care so much about these blooms, not just in Chautauqua Lake, not just in New York State or the US, but truly around the world. This is a global issue that many, many lakes are experiencing today. So let's think about what are some of the factors, what are some of the controls on algae growth, algae and cyanobacteria growth? What are the things that drive bloom events? They're primary producers, they perform photosynthesis. So a lot of these variables are pretty similar to what you might see for land plants, especially the big three: light, temperature, and nutrients, okay? And when I say nutrients, we're talking about nitrogen and phosphorus, okay? Those are the two big players when it comes to controlling algae growth in our freshwater lakes. Now, this might seem sort of straightforward. We know these things, right? But one of the tricky aspects is that when we look at these algae that are, these cyanobacteria and algae that are growing in the lake, this is not just one species most of the time. There could be tens or hundreds of species that are active at any given point in the season. And each one of those has different requirements. So just like when you look out in the landscaping, some plants are more shade tolerant, some need more nitrogen rich, right? And some like a certain amount of temperatures, right? Some are early season and some are late. And so the same sorts of rules apply to algae. They have different combinations of resource needs. And that's a lot to sort of weigh in and figure out how do all these pieces come together. So this is sort of the foundational piece, but there are other things that are more indirect that also affect how algae access these resources. Things like the internal mixing. right? The physics below the surface of the lake are moving algae around and moving resources around to the point where sometimes they are changing their exposure levels and what's available to them in the lake system. In addition to what we would call the abiotic or non-living factors, there are also a whole host of biological interactions that are also at play, just to make it more interesting. Grazing can occur where we have zooplankton, or the next level up in the food web, that eat the algae and cyanobacteria, right? Changing the population structure, sometimes selectively choosing the salad over the Big Mac, right? And so they're changing how those organisms are growing. And they can also be in competition with each other at that same level, right? The algae and cyanobacteria are trying to do the same job and they're fighting for the same resources. So competition will typically lead to somebody being more dominant at one point, but then eventually getting pushed out, right? Because somebody else is better at doing that job under those conditions. So in New York State a few years ago, Governor Cuomo had his HAB initiative, and we identified through that process of exploring data for the 12 priority lakes across New York State, including Chautauqua Lake, identified 4 main risk factors for algae blooms for lakes in our state. Phosphorus, zebra mussels or Jasinin mussels, the length of the lake, and then the compass orientation, how the lake was sort of configured compared to prevailing winds. And we have 3 of those risk factors here at Chautauqua. So it's no surprise that we're susceptible to blooms based on that analysis. So we know these big swings, right? We understand that these are the important players, but trying to figure out exactly how these pieces affect algae and cyanobacteria, how they interplay off of each other, is like trying to solve a Rubik's Cube where the little colors are changing because they're connected to another Rubik's Cube over here that's connected to another one over there. And then somebody else has been playing a Rubik's Cube for 10,000 years and you don't know the story of how it all got there and it's all interconnected, right? So when we say we understand the basics, but we don't necessarily know exactly what triggers a bloom at any particular time, right? The exact set of variables that come into play are really hard to track in the real world when this stuff is happening below the water surface with microscopic living things, right? So it's a big question in terms of how do we start to tackle this? How do scientists approach this problem? And so I wanted to spend some time here, and sorry, this is a little bit small, but I'll walk you through all of these ideas, but talk about what scientists use to study algae and have events. And truly, there are a range of things that we could select from, right? I want you to really realize that there is not one best way to answer the question of why did the bloom happen, right? There is not one best tool. In point of fact, they all have their limitations. I tell my students, there is no free lunch in terms of measuring this. There is no golden ticket. There is no one way that will tell you everything you need to know. So you have to think about as a scientist, what are the tools that I have available to me? Which one is best suited for my question? And logistically, what's feasible, right? In terms of the challenges of field work and getting materials back to lab and analysis time. And so we have to sort of think about all of the different perspectives that each of these tools might provide and then select from the menu, right? So one thing that you might have heard fairly frequently is chlorophyll, right? Does that sound familiar? Chlorophyll concentrations? Yeah, good. Okay, excellent. You're good, right? I can go? Yeah, okay. So chlorophyll is the primary, chlorophyll A in particular, is the primary pigment that algae and plants use to perform photosynthesis, taking energy from the sun, using that to build carbon chains from carbon dioxide and put that into sugars, right? And so chlorophyll A is the main workhorse. And so we can measure the amount of pigment present, and that represents the amount of algae present. So more pigment, more algae. And we can call that also the standing crop, right? What's present in this moment at this time. And so there are many ways that you can possibly measure chlorophyll, right? You can collect lake water and then filter it, which is what we see here. There's a little filter here, all that sort of, this is a little bit more golden brown than bright green, but that's because it's a different group of algae than we're used to seeing. So that sort of color represents all of the pigment that's present on that filter. We can then take that filter, pull the pigment out and run it on a machine in the lab and see how much pigment is present. You can also take a sensor into the field or use one in the lab, like this one here. There's an optical sensor here that measures the fluorescence of those pigments in the field, right, in the lake itself. So you take your sensor and you lower it down through the lake and measure what's there. And there are other ones that you can also use this in the lab as well. So sort of more of a live representation of what's happening. There are more tools, and I'll talk a little bit more about this later, but tools that use remote sensing, so satellite imagery, for example, where you get information about an object without touching it. And so satellite imagery uses the optical properties of what it's scanning and a lot of math to calculate then the amount of chlorophyll that we suspect is in there based on the properties it's collecting from its sensors. So chlorophyll, and again, this is a very short list of all the different flavors of chlorophyll analyses. But chlorophyll is a bulk measurement, right? It gives us really good quantitative information about how much growth is present in the lake, right? But it doesn't tell us the species that are doing it. It doesn't tell us who are the players on the stage. So we need some other tools if we want to know more about who's being active, right? And so one of those tools might be cell counts on a microscope, where you collect the water, you bring it back to lab, and then you actually go through here and you look at all these individual little cells and you count all of those and you identify them to as far down of detail as you can. This gives us a lot of information on who's present and who's active, but you can imagine that this is very time consuming and requires a lot of expertise. And honestly, especially for cyanobacteria, it's difficult to differentiate one little green circle from another little green circle. And sometimes when we preserve them and bring them back to the lab, they change, right? The act of preserving them changes how they appear to us. So it's tricky, right? It's a great tool, but it's not necessarily the whole story all the time. You can also, another way to approach this is to say, well, I want to know the rate of growth, right? I want to know how fast algae are growing. And you can measure the rate of photosynthesis by how fast they take up a particular chemical. And we can actually use radioactive isotopes, tag that carbon, and then watch how much they bring into their cells. This is the most precise way of measuring algae growth rate. Again, not so easy to do, right? Radioactive isotopes require a lot of specialized skills and they're highly regulated. And I can tell you from personal experience that folks don't always love it when you have big radioactive stickers lakeside while you're working, even when you're following all the rules. It's a challenge, right? So it's a great tool, but it's not always the most appropriate to deploy, right? Like it worked for me pretty well in North Dakota where there were like 3 people. I would not do it here. There are way too many people. It'll be way complicated. So another tool that we use often in assessing HABs is this idea of toxin concentration, right? And this makes a lot of sense because that is the most important thing from a public health perspective, right? We want to know, is it dangerous for people to be on or near the water? So measuring toxins seems like a no-brainer. The challenge is that our analytical process is slow, right? It just is. This is not easy to do. There's also not just one toxin. There's like multiple flavors of one toxin, right? Microcystin has like 4 or 5 or 6 different types of microcystin that might be there. And that doesn't even talk about all the different other categories that we know about, right? If we don't know it's there, we don't even know to look for it. So toxins, if you want to know more about it, you have to measure separately for all of those different toxins. It's a very specific analysis and it's slow. So algae blooms are notorious for changing their toxin production relatively quickly. So you can go out at noon, collect your water sample, send it to the lab. You can overnight it right there, and it'll still take you 24 to 48 hours, if they're fast, to get you the data back. And an hour after you collected the sample, it might have started producing toxins. So you might get a negative from the lab, but it's a problem, right? So it's a challenge. And that's one of the things that there's a lot of really cool technology that's evolving right now. And so I think we're compressing that time and we're getting faster, but it's just not easy to do, right? It's a very specific molecular analysis that has to be performed in a lab setting. One tool that's also come on in the last 10 years or so that's been really exciting is the use of eDNA or environmental DNA, meaning that sometimes we can't always see everything that's present, but a lot of times it leaves traces of its DNA behind, right? It's genetic material that identify it specifically to that organism. That's left behind. Even if you can't see the thing that made it, it can leave its DNA behind. And we can go in, extract that, amplify it, and measure it, and connect it to these amazing databases that tell us who is present in the system. So eDNA allows us to do more genetic tracking. And again, a lot of work involved, preservation matters, but it does sometimes give us a closer look when we can't tell one green circle from the other, right? So it can help us differentiate the species that are present in many cases. The last thing I want to mention is sort of on the other end of the technology spectrum, the Secchi disk. That's this little black and white disk here, right? Super high tech. It's literally a plastic disk with black and white paint on it. That's all it is. You put it on a measuring tape and you lower it over the side of the boat. And when it disappears, that's called your Secchi depth. It's about 10% of light remaining in the lake. And it's a really simple, inexpensive tool. It's easy to train people to use this tool. And most of the time in many systems, algae are affecting the transparency of the system more than other things. It's not the only thing that can affect water transparency, but often the algae growth is connected to that. So there's some limitations, right? It's not super precise in terms of specifically algae growing, but it's very cheap. It's very easy. You can do it all the time. There's no lag time for lab weights. You get your number right there. Yeah. So from this lengthy for you, I'm sure, but short from my perspective list of things that we could use, there are all these different potential pros and cons, right? And so when you're thinking about your question, we as scientists have to weigh which of these tools or which combination of tools will help us get to where we want to go, right? What is most appropriate for us to use? And so not everybody uses the same tools all the time, right? And that's normal in science. How do we make those choices? Well, it's sort of like the real estate mantra. We're going to borrow from that. So we're going to say scale, scale, scale. We have to think really carefully about the question we're asking and the scale in time and space that we want to work in. And so blooms are what I would think of as a macro issue, right? You look at the lake, it's green. It's a big deal and you can see it. You don't need much special to see it when it happens. It's right there, right? And so it's a macro issue. But what's driving that, what's causing that is all micro, right? The algae and cyanobacteria growth, those are cellular, right? Stuff that you cannot see individually with your own eye. And all the other factors that drive that, right? The light, the temperature, the nutrients, the grazing, those things vary in scale too. So they are not, some of them are macro, some of them are micro. So figuring out this question of what causes this, right, means that you have to detangle all of these things going back here. So this Rubik's Cube in motion, right, is doing this in part because lakes are always in a state of flux. They are constantly changing, right? The lake 12 hours ago is not the same as the lake is today, right now, right? The light, the temperature, these things are different, okay? Some things are stable, but some things vary quite a bit, even on a daily scale. We talk about seasonally, the lake in July is not the same as the lake in January, right? And you could have this go back and back and back to the origination of the lake about 15,000 years ago. When the glacier sheet receded, the lake then when it was formed is not the same as it is today, okay? So in thinking about the time scale, we need to be aware of that as scientists and make sure we're blending different temporal context. The other thing that's interesting about lakes is that spatially, they are also very different from the surface of the lake vertically, right? From the surface to the sediments and from the shoreline into the open water. So what you have happening is truly a 4D system, right? We have 3 dimensions plus time, and it's always changing. So for us as scientists, this is very cool, right? This is very interesting, and it's fascinating to try to solve this puzzle. But when it comes to understanding it from the community aspect, when you're affected by this, right? When you can't go boating or swimming, or if it doesn't smell great when you're trying to have Sunday brunch with your family on your deck, right? These are things that are kind of hard to connect back to these puzzles that I find interesting as a scientist, right? So when we tie this together and we're trying to understand this big problem, we are really, as scientists, coming back to this question of how do we study it, we're trying to figure out how to deploy the right tools at the right scales and think about how we blend multiple perspectives. So we usually use a combination of things to answer a question, right? I rarely go out and measure one thing at a time. I usually use lots of tools that I have to gather as much information as possible. So our more traditional methods of lake sampling rely on us going out into a boat, getting to a representative spot in the lake, maybe 1 or 2 places, maybe 3 if we're lucky, right? And collecting the water, pulling it back to the lab, and doing all of our analysis, right? And it gives us a lot of really detailed information. But because of the investment of time and labor it takes to analyze 1 day of work, we can't do that every day in all places, right? It's just, you can't do it. There's not enough person power available or enough analytical space available to complete that in that way. So traditional methods give us a lot of really good quantitative numerical information. What we can do is blend those techniques with more recent technological advances, things like the remote sensing I mentioned with satellites and drones, where we can increase our spatial perspective by being farther away. We don't get the same information we can get by contacting the lake and grabbing the lake water, but we do increase our spatial perspective. We can see more of the lake at once. We also have some amazing advances in sensors that can be put out in the lake and left to collect data for us. And oftentimes they can do this at very high frequency intervals, right? Meaning that they're collecting data sometimes in the minute to minute scale and sometimes multiple times a second. So this sort of information is very powerful. And we need the computing chops to back that up, right? Like, in order to do something meaningful with 4 measurements a second for like 4 months, you need some real data heavy analytical power, right? Like it's not gonna like happen on my laptop. So you have to think about, what do you do with the information after you collect it? Additionally, the rise of the improvements in communication networks means that for some of these high frequency sensors, we can actually watch the lake in near real time. And this is one of the things that gets me most excited about working with the Jefferson project, with their platform buoys that are coming next week, is that they have wonderful sensors, they have great communication, and they have the modeling power to do something with that data meaningfully. But it takes time, right? Even though the data collection is fast, the analytic part is time consuming. And so all of these tools in the technology, this newer technology realm, give us better vertical and horizontal coverage, right? They tell us more about what's happening in different parts of the lake and in more times, right, during the lake's active periods. The true key to studying lakes, honestly, because there are so many things to know and so many things to understand, the true secret to this, the secret sauce, is collaboration, is working with different people who have different expertise and strengths and learning new techniques all the time. And when you package it together, it allows us to advance our understanding in some really amazing ways. So it's been an exciting past few years here at Chautauqua, having so many more academic scientists around the table active at this lake. It's a very cool opportunity. I've been here for a while and sort of doing my own little thing, but it's really ramped up, and it's been great to have more people, because we can answer much cooler questions, right, with more folks around the table. So that's my overview of blooms and what we know and how do we study them. Okay, I want to shift for the last portion of the talk to Chautauqua Lake specifically and what's happening here now. I'm going to give you a brain break and get some coffee. All right. So in terms of Chautauqua Lake, just to review, so we're all on the same page again, it's about a 13,000 acre lake, or about 53 square kilometers. It's 27 kilometers long, 3 kilometers wide. So it's a long, skinny system. it has sort of this dual basin configuration with a slightly deeper north basin and a shallower south basin. In general, though we're learning more about how the water flows from north to south, right? It exits at the Chautauqua and goes down to the Ohio and Allegheny. It's relatively shallow for its size. When I first drove over Chautauqua Lake, I was like, oh, that thing's got to be 200 feet deep. No, right? It is not the case. And so for its surface area size, it's shallow, right? A max depth of 76 feet in the deep hole in the North Basin. But most of the North Basin is 30 to 35 feet, kind of flat bathtubby situation, about 14 to 15 meters, right, on average there in the center portion of the lake. It's because of its shallow nature, it has a relatively short retention time or flushing rate, meaning that it's about 2 years from the time a water molecule enters the lake and then exits. So many lakes this size that are deeper have retention times on the order of hundreds of years, right? So this is very short given the size of the system. It's shallow and we get a lot of rain. So it makes sense that it's flushing quickly. It's classified as eutrophic or mesoeutrophic, meaning that it has high productivity, a lot of plant and algae growth occurring in the system. And so we do have frequent persistent HABs. As you know, we use Chautauqua as a tourist hub for world-class fishing events, recreation, and drinking water. So there's a lot of complexity in this system. And from a scientific perspective, that's very interesting. But it means that we have a lot of things to sort of weigh and measure and keep in mind when we're doing our science. I do want to give you a little bit of backdrop in terms of the longer term trends at the site in terms of its water quality. We are incredibly fortunate at Chautauqua. Has anybody heard of CSLAP? Mark wins. No. CSLAP is the Citizen Statewide Lake Assessment Program. It's New York State's volunteer monitoring program. And so lakes can opt in. And we are very fortunate to have had Doug and Jane Conroe collecting data at Chautauqua Lake on a volunteer basis every 2 weeks in the summertime. And we have a record going back to 1987 for Chautauqua Lake, which is great. Most places are not lucky enough to have this kind of data. So it's an amazing set for us to look at. If you ever want to look more at Chautauqua Lake's records or other systems that participate, the older reports are available on the NYSFOLA website, the New York State Federation of Lake Associations. You can find the old reports there. And they've just moved everything that's more recent to the new CSLAP data dashboard that's much more interactive. And you can kind of click on things and explore it that way. So I encourage you to take a look at these resources if you haven't done it yet. So I want to show you some data from last year, from 2022, for lake water parameters. And we're going to look at the water transparency, that SECI disk that I mentioned, the black and white disk, chlorophyll, and then phosphorus concentrations, okay, in this panel. And what you'll see on each of these is that we have, it's a little bit small for you, sorry, but for water clarity, we go back to 1987 when the record started and then comes up here through 2022. And in this case, we have the depth. This graph is a little bit funny. So we start at the surface as 0 depth and going down to about 10 meters here. Okay. And so it's 10 meters down here. And so when we have these disk measurements, right, a deeper reading means that it's more transparent. It is clearer. And so this is associated with lower productivity lakes or oligotrophic lakes. Lakes like Chautauqua land more in this mesotrophic or moderate productivity, or eutrophic, highly productive, that lots of plants, like the green lake, right? So you sort of have green up here and blue down here, right? You can see that we kind of ride this line here between mesotrophic and eutrophic for the history of this record, right? So we're sort of hovering around, around 2 meters of water transparency. So a little bit around 6 feet. Additionally, for chlorophyll, that measure of algae biomass, we have our, again, record going back to 1987, going up through the present, and then our summer chlorophyll concentration. And so what you'll see here is that we land in this eutrophic zone. The threshold is right here if you can't see it. We tend to land more solidly in the eutrophic zone for the chlorophyll. Okay. And then I'm also including phosphorus here. phosphorus is one of those key limiting nutrients, right? That is really focused on with lake issues because it tends to really spur a lot of cyanobacteria growth. And so when we look at phosphorus growth, phosphorus concentrations, excuse me, in the deeper waters, we actually get some deeper waters and surface waters here, but you'll see in the deep and surface waters that we kind of tend to be, at least aside from the first few years of the herd, into the eutrophic zone again, right? It's a bit small, but what I do want to point out is that, I don't know about you, but I hear a lot of stories, and sometimes people say the lake is as good as it's ever been. And then sometimes people say it's as bad as it's ever been. Yeah, like I hear really mixed bags. It always depends on who I'm talking to and where they live. So, and their experiences, right? But what I want to point out here, when we look at the lake water quality for Chautauqua Lake, in the best data set that we have going back for the most amount of time, the most time context, you can see that the lake is actually relatively stable, right? We don't have massive upward trends in blooms. We don't have major changes in transparency. Even phosphorus is high, but stable. So I think this is I want to bring this back to this message of what do we really know? And there's reasons why this data set may not capture the whole story. There is a reason why somebody in a different part of the lake might say, no, it's worse than ever. Because these are that sort of traditional method where we go and we collect from 2 locations in the lake for a 13,000 acre lake. That's not, we're assuming that that's representative, but it may not be, right? Because we can't sample everywhere all at once. So I do want to point out that we have variability, right? It goes up and down each year, but the trend overall is fairly stable, high, but stable. Okay. I wanted to show briefly just the toxin data. The toxin data is something a lot of folks are interested in. And when we look at the open water toxin levels for microcystin versus the shoreline toxin levels, also for microcystin, you'll notice that most of the open water samples, this is from 2021, most of the open water samples were below the recreational level of concern, even during bloom events that were reported. But the shoreline toxin levels in 2021 were almost all above the recreational level of concern. So I didn't show the 2022 data because there wasn't shoreline data for 2022. So I wanted to show you the sort of spatial differentials, right? And how where the algae is sort of initiating and growing versus where they're accumulating might have different measured variables, right? The way we assess the bloom condition, really, we have to be careful about that as scientists, right? There were 49 HAB reports, voluntary reports in 2021 and 33 HAB reports in 2022. Those are responsive, so it depends on who sees it and calls it in. So I wouldn't put much stock in higher versus lower. There's just a lot that's happening, a lot of reports that are called in for blooms. So even though things are stable, we have a lot of concerns, right? And so it's important that we as scientists are figuring out how to tackle this. So in the last 5 minutes or so, I want to tell you just a little bit about what my research team does, and I'm happy to answer more questions in detail during the Q&A, but I'll just discuss 3 little pieces that we're working on in the most recent past. And so if you've heard me talk before, you've heard my big question of how do lakes respond to different kinds of environmental change? That's my big umbrella question. We are mostly an undergraduate institution at SUNY Fredonia, so we're primarily driven by the undergrads and a small master's program. And I always need to stop and give my students credit because they push me to ask really cool questions. The research that we do is driven by their ideas and we work together. And so I have to tell you that my students come up with things that I would not have probably done, and it always turns out really cool. So I have to give them a lot of credit for this and all that work that gets done. And so we use drones, we perform experiments, we look at lake history, use temperature, high frequency sensors, we participate in routine monitoring, and we collaborate with folks from Bowling Green, the Jefferson Project, and other academic teams to do all sorts of things related to stream sampling and experimental work. So we work a lot with the other people that are here as well. So I don't have time to talk about all of it because I've already probably melted your brains a little bit So I promise not too much longer. But I'm just going to highlight 3 little pieces to give you a sense of exactly how do we deploy these different tools, right, for how we study algae. The first is using drones to study blooms from a more spatial perspective. And so in science, we know the big triggers, right? But we don't really have a sense of sort of the bloom life cycle. How do they originate, move, and then die off, right? And so we need that better spatial data available to us. And so what we've done through a grant funded by Corning Incorporated is to fly a very nice drone with a very fancy hyperspectral imaging camera over top of the shoreline of Chautauqua Lake and pair that with ground truthing water samples below that. The hyperspectral camera gives us way more information about different wavelengths of light and optical properties that you don't get from a standard camera, which helps us when we're trying to understand who's present in the lake, right? Try to separate out the algae from the plants. Yeah. And so we're connecting these things right now. We have a new colleague at SUNY Binghamton, Dr. Jason Bobin, who just got an Air Force Research Lab grant to continue with this project. So we have some preliminary data, but we're still working on connecting the water samples with what the camera is reading from the drone. So early stages, but very exciting tools. Plus toys are fun, right? What we haven't talked about yet is the fact that a lot of our tools are mostly observational, right? When we go out to the lake, we are seeing what's happening. And so a lot of times we see correlation, we see relationships, but we don't always know that it's a causal relationship, right? We know that A and B go together, but we don't know that A causes B unless we do an experiment to control all these other things that might be affecting our results, right? Affecting our observations. So one thing that my lab team uses a lot are these microcosm experiments. They're basically fancy science Ziploc bags that have lake water in them and then have different amendments, different nutrient additions or other things we want to explore. And it's a very flexible design. And so it allows us to, in a short period of time, in a very controlled way, test specifically what causes algae to grow under these conditions. And so we've most often done this, we've done this repeatedly at Chautauqua. We mostly use nitrogen, phosphorus, and then a combination of those. So we're primarily looking at the nutrient controls on algae growth. But we get really interesting responses. So what this looks like at the end is if we have our chlorophyll here on the left and our treatment on the bottom, you'll notice that our control treatment grows here. Our phosphorus, when we add phosphorus to it, doesn't always give us much of a response, right? What does is nitrogen. And even more so when we combine them, right? So while much of the focus in lake management is on phosphorus, at Chautauqua Lake, we also need to be paying attention to other nutrients that might be important. And I could do a whole other lecture on why we focus on phosphorus, and it makes sense, right? But it's sort of a default, right? And that works for a lot of systems, but for Chautauqua, we know that both nitrogen and phosphorus are important. And also I'll mention the North Basin is not the same as the South Basin. They're not the same if you do it June, July, and August. And they're not the same from 2016 to 2018 to 2019. So they're different in a lot of ways. So it's, again, interesting for us to solve, but complicated. And this is a helpful tool for us to really drill down. Speaking of going down, we also in my lab use lake sediment records to look at the lake history to expand our time perspective. And we can do this because lake sediments contain an archive of the past conditions. We mostly look at these things called diatoms. They're not a harmful species, but they do have a glass cell wall that preserves really well in the sediment. And they're very sensitive to different kinds of environmental change. So they tell us a lot about what's happened before at the lake. They're also very pretty. So I like that. We do have pilot cores from North and South Basins. We have good indications for preservation of diatoms, some changes in the recent past, which we aren't sure exactly what it represents, but the lake is shifting just in the short cores that we've gotten so far. I mentioned eDNA earlier, and one of the things that we just did this spring that's very exciting is we looked in the sediment records to see if we could find cyanobacteria eDNA, if we could find their DNA present, and we've gotten some hits. This is very important because cyanobacteria don't leave visible things behind, at least not reliably for us to look at. So anything we know about cyanobacteria only goes back as far as like the recent past, right? We've got maybe 20 years of cyanobacteria data. That's it. That's not a lot of time in the lifespan of the lake. So eDNA and combined with sediment cores is a really cool tool that we're working on. So overall, we know that blooms are problematic. We've had a chance here to talk about those key drivers and control, but also I hope you understand a bit more today about why scale matters and why scientists choose these different tools that we have available to us to answer particular questions in space and time. And it's a lot about, like anything in life, it's a lot about balance and figuring out how do you put these pieces together in the way that leverages the strengths, right, and shores up the weaknesses of your different approaches. And so we're working on this. So I have a great team of students that are very active, and again, very exciting to have these wonderful collaborators present at the lake. If you do see a HAB, this is just my little plug. If you see a HAB and you're not sure what to do, the DEC has excellent resources available for you from the public. You can submit this through the NIHABs reporting system. You just go in, you can just Google it, Google NIHABs. Take 2 pictures on your phone and upload the GPS points. Somebody from the DEC will then look at the picture and assess whether it needs further investigation. And that's very helpful because it helps us monitor from a public health standpoint and scientifically to know what's happening with the system. Their mantra, the DEC's mantra, is know it, avoid it, report it. You cannot tell when you look at a bloom if it's toxic or not. So the best case scenario, especially for kids and pets, is to keep them out of the water, right? That's the safest bet. I can share these later if you have any other questions, but the CSLAP reports are available there. There's also the HAB action plan for Chautauqua Lake available through the DEC and a wide array of HAB information that they've put out. There's wonderful brochures and pamphlets and videos and things. So our New York State DEC has done an excellent job of providing resources to the public to educate yourselves. So I encourage you all to check that out. Many folks to acknowledge, lots of past students, community partners, funding resources, and in particular, thanking my students and the foundations that support their summer research because they're out busy today on the lake working on stuff. So with that, I will take questions.
So please come up to the microphone to ask your questions so that we can hear the question as well as the answer. We're trying to monitor also, if you're zooming in, we're trying to monitor the chat function, the chat room, and I'll ask Mark to come up once in a while with the questions coming in from the chat room. But this was a great talk. Please ask your questions.
Thank you for a fantastic talk. I had just 2 very basic questions.
Sure.
As a scientist but aquascience novice. When the term bloom is used, most people think of flowers blooming. Why is bloom used in this context? And the second question is, when we talk about algae, we think of plant. When we talk about cyanobacteria, obviously a bacteria. What is the interrelationship between the plant and bacteria?
Okay, yeah. So great questions. So the bloom question is really, I don't know the exact answer. I can guess that it's really about the explosive rate of growth, right? And when I see this picture, it makes me think of a bloom, right? Sort of exploding out from a central point, right? It sort of kind of grows up and expands. So a bloom in this case is really referring to that rapid expansion of growth. So that's a good connection between the flowers. I'm not sure exactly any like language people in here that would know that, maybe not me, but it's a cool connection. So it's all about that growth rate being really fast. The other question was the relationship between algae and cyanobacteria and plants. Yeah. So cyanobacteria were our first photosynthesizers, oh boy, 2 million years ago that started doing this job for us on the planet. And so they were the first ones who were doing this job. They were the simplest of cells, right? That were doing this job of taking energy from the sun, converting it into sugar, and then additionally, as a happy byproduct for everything else that came later, producing oxygen, right? And that really was a game changer for our atmospheric composition and then all life that came after that. So we have a long trajectory to be thankful for cyanobacteria in terms of us being here. But later so the bacteria are one separate branch of life, right? The prokaryotes, they don't have internal cell structures, right? And then there are the eukaryotes, which are another major branch, a major domain of life. And that's where the algae group comes from, algae and plants and us, right? Everything else that's not bacteria or another form called archaea, everything else that's more complex multicellular life is all in this category over here. So actually you and I are more closely related to the algae in the lake than the algae and the cyanobacteria are together. Yeah. Yeah, science lesson for you all today. And so it's interesting to think about it that way. The land plants originated, we think, from the algae group and a group called charophytes. And so we think that that was probably where they came from, right? So the life originated, we had aquatic life that was originated on a planet, and then things moved on land. So it likely originated from that group.
So just a quick follow-up question. Are the cyanobacteria then on the algae, or is it part of the algal structure?
They're separate. They are separate individual life forms that are living in the lake. They interact, right? They interact, they compete with each other. Some algae and some cyanobacteria like to grow attached to things. And so sometimes they do live on plants. Algae and plants have a complicated story, right? So they might be in competition with each other. They might be supportive or mutualistically benefiting each other, or they might not care, right? They might not really do anything for each other. So it depends on the exact set of species you're talking about as to what that relationship really looks like. So there isn't like a single answer to that question in terms of the algae and plant relationship, but often they're competing for light and nutrients, right? So they can be in competition with each other.
So in Lake Chautauqua, for example, do you find cyanobacteria just freestanding, free living, and then algae free living, and sometimes the cyanobacteria are attached to the algae?
Sometimes they can connect. So there are sort of macro forms of green algae, and you might see it out probably around now, and certainly in the past month. There are sometimes mats of green algae, filamentous. If you ever see those sort of like felty looking bubbly mats of algae, they look really nasty, right? They are just green algae filaments. They're safe. They're fine. They look really funky. But in those cases, those sorts of mats can sometimes contain a whole host of other life, right? They can provide sort of resources and a home base, but it depends on where the algae and cyanobacteria prefer to live. Sometimes different species prefer to live attached to something, right? And that's how they survive. That's their growth form. But many of the times, what we're talking about for blooms are the plankton, the free floating cells that are out in the lake, mixing up and moving around. When we have a bloom form, sometimes they form a scum because cyanobacteria have like little life preservers. They float, they regulate their buoyancy up and down to get more light or to get more nutrients, and they move around. And so eventually what happens is many times they're floating at the surface to get as much light as possible, right? And so they might look like a solid piece, but it's just all the individual cells that are clumped together.
Thank you.
Yeah.
Two questions. One, thinking about municipalities,
Yeah.
how, and the challenges of testing that you guys experience.
Yeah.
How do cities test for water just on a regular basis?
For drinking water?
Yeah,
Yeah, or lake sampling.
drinking water.
Drinking water. Yeah, so there are different regulations by the state, and I'm not super familiar with the details, but a lot of times they'll test at the intake and then at their processed water, right? Depending on the steps that are required. And I don't know if those are daily or weekly, but there's routine regulations that are in place to make sure that that drinking water is safe. You can always step up the sampling intensity, but that comes with logistics and costs and all the pieces that go with that. So there's a balance when you're monitoring. You want to make sure you're keeping people safe and you're monitoring frequently enough, but not so frequently that you're doubling your costs for your community, right? So you're kind of weighing all those pieces. I don't know specifically what the CUD does here for Chautauqua. That's a great question, exactly. But I don't know if it's daily or I don't know.
I think we have a representative from Chautauqua Utility District here. If they want to identify, if he or she wants to identify themselves and answer that. We really like the idea of scientists and people who have to implement things talking to each other. So we're glad to see this.
Governor Cuomo's HAB initiative started a whole range of new
Yeah.
testings. Most of them start with identification of a decent sized bloom, not just a random
Right.
spot here and there. And what we've been doing since probably 2018 is at least weekly, sometimes bi-weekly, take a raw sample coming from the lake and take a entry point sample coming out of the treatment plant. It goes to the state lab, either Wadsworth in Albany, or they've been farming it out to the Erie County Health Department. And through a very convoluted chain, I eventually get results sometimes within 5 to 7 days and sometimes longer.
Yeah.
So if they do find something, I don't hear about it for a week.
Yeah.
And neither will anybody else, because we're still treating the water and
Right.
filling it out. We have never yet had any show up in our entry point water. We do use potassium permanganate, which is a mild oxidizer. It started out as zebra mussel control,
Okay.
but it also does a good job of killing most all of the algae,
Okay.
and it does help take the cyanobacteria out of its cell structure and it falls apart. But you have to have the proper dosage rates in order to make that work the right way. And it varies depending on how high an algae bloom there is and how long it's been going. What our practice has been is that if there is an algae bloom sighted in the North Basin, we start monitoring hourly, looking for turbidity, seeing how dirty the water is. If the turbidity starts to rise real fast, or if it goes immediately to a high number, we just shut down. And usually within an hour or two, it's floated by. It just moves around out there. We have had stretches where we had to do that every day for 2 weeks, where you shut down for a while and work at it. But a lot of the time it is mid-August to mid-September is when it's at its worst. And there's not as many people and we don't have to operate as many hours a day. So it's easier to do.
Yeah. Thank you.
Yep.
Thanks.
Other question was, are there any pristine lakes, either in New York or even in Canada, that would offer a different baseline insight to
Yeah.
blooms?
Right. So the word pristine is always really interesting because we are having an impact even on really remote systems. So there are many, there's 305 million lakes on our planet by estimates. And they come in all flavors, all shapes and sizes. So one of the things that's really powerful in ecology is comparative analysis, right? Not just looking at Chautauqua Lake, but looking at as many lakes as possible to understand what's going on. One of the 12 priority lakes that was established is Lake George from New York State. In terms of a large system, it's not the same as Chautauqua, right? It's very different, but at the time of the selection for that, it hadn't had any visible surface blooms. Now they've had a couple of very short-lived ones in the last couple of years, right? I think last year was maybe their first reported bloom. And it was gone in like 2 hours. So we would think like no big deal, right? But what Lake George provides for us is that context. What is different about Lake George? Why don't they have blooms there? Why do we have them here? And it's one of the really exciting things about working with the Jefferson Project in terms of stretching the scale of what we have here at Chautauqua compared to other sites. So they have this really amazing, technologically highly instrumented lake at Lake George, and they're bringing that system here to take all those lessons they've learned to understand more about our processes at this site. I think that it's extremely powerful to look at other places and learn other lessons from other types of systems. We did the math 20 years ago. hiking up mountains in Montana and Wyoming to study the lakes up there. And you could call those lakes pristine, but in point of fact, they're not, even though they're sitting up at 12,000 feet and there's no trail and there's no buildings. There's like the one lonely angler that like walks up there a couple times a week and goes and fishes, right? But that lake is being affected by climate change, and it's being affected by atmospheric deposition. There are air masses that are moving over, dropping nitrogen in that lake and causing increases in certain species of algae. The algae are so sensitive, we cannot detect the chemistry difference in the lake, right? Those changes are so subtle. The algae are responding before that. We can see a change in the community structure of the algae that are present before we can detect it chemically. So the biological signals are there if we know to look for them. So I started working out there. And then I did work on the Plains Lakes and actually inland salt lakes in North and South Dakota in the Prairie Pothole region. I've worked on lakes out in Maine. I worked on lakes in South America, in Argentina and Peru. And when you put and I grew up on Lake Michigan, right? So I'm no stranger to the Great Lake systems either. But every time I look at a new lake, it's absolutely fascinating how different they are. It's like a fingerprint, right? And so, but they're driven by the same big things. And so I think that's really powerful to remember that Chautauqua is a part of that bigger story. And there's lots for us to learn. It's all going to operate uniquely at our site because of its specific features, but we can learn a lot from other sites and try to understand why it is the way it is, right? So it gives us those extra flavors, really build the story for us.
I was going to ask a question, but we filled our time, so I'm not.
You can ask, I'm good. Sure.
You don't have to run off, though. I really do appreciate that we had a friend from the utility district here, and we're going to try to do more. One of the beauties of this process is to link science and our everyday lives. And it is amazing how many aspects of science affect us every day. And we're lucky, it just skims over us and we don't even think about it. So we're glad that you're here. I love maps. The fact that you look at a map and you see, wow, this lake is north to south, it's two basins, the wind's blowing mostly west to east. And what does it all mean? And does it mean the same thing every day? You know, we have a 1 million gallon water tank. So it's like that water's not going straight into your mouth from the lake. It's being processed and saved. And so don't worry about that. But I want to thank you for being here. That on the back table, there is a list of all the programs for all year. All summer. Please come back. As I try to say every week, it does take a little bit of resources to do this. There's a basket. If you feel so inclined, put a Ben Franklin or a Thomas Jefferson or an Alexander Hamilton, anything in that basket, we'll appreciate it. And we'll see you next week.